Hello. Well, right now what we're going to do is explore the adaptive immunity. This would be lecture 11 in medical microbiology. Now, just as a review, you've got to keep in mind that we've already dealt with the innate immune system, which is the first level and second level of defenses. Now what we're going to be dealing with is the third line of defenses. Now, the difference here is the speed, or I should say, the timing of its response to the initial infection, as well as the specificity of the particular pathogen. To give you a review, the adaptive response is the body's third and final line of defense. Adaptive responses are called into action when the innate first and second line defenses fail to contain the threat. Adaptive responses take longer to mount than innate. Innate can happen very quickly at the time of infection. Usually, though, adaptive take a few days to get really revved up. Adaptive also is focused on the specific antigen. And this gets us into that term of immunological memory, response to the same antigen upon subsequent exposure. Here's one little clip. If you had the flu and you developed the adaptive responses to the antigens inside the flu virus, but comes another year, you have a new flu virus. It's actually the same flu virus, but it's mutated, has some different antigens on its surface. In both exposures, you would have the innate responses kick off in an attempt to prevent the infection or interrupt it, although they would not necessarily be successful. The adaptive in the first season has been taught to look for a specific line of antigens. But now the antigens on the exterior, let's say, of that influenza virus have changed because of mutation. So the adaptive responses are not going to be as effective, if at all, play a role in defense in this next season's flu. And that's why we basically make season to season vaccines against the flu. When we talk about the immunological memory, though, this is the response, as I said, to the same antigen upon subsequent exposures. Now, the great thing about this is that, for example, if you've been exposed to a particular disease, let's say chickenpox, for example, and the next year and the year after that, you don't get chickenpox, even though you might be in front of people that have chickenpox actively. Your immunological memory will be active enough, and you'll have memory cells that will provide a defense against the next season or a year or two later, that same chickenpox virus exposure, and you won't get chickenpox the next time. Now, as we deal with adaptive responses, they can be divided into two primary responses. Cellular response, this is referred to as T-cell mediated immunity. And humoral response, this is referred to as B-cell or antibody-mediated immunity. Okay, so humoral response and cellular response, we're going to be going through a lot of this in the up and coming slides. Now, when we talk about the adaptive responses, they progress through four main stages. You're going to see this both for the humoral as well as the cellular responses. First, the antigen presentation. In each case, dendritic cells and certain other white blood cells will act as antigen-presenting cells. Keep in mind, I'm going to be using APC. It's much quicker and faster than just saying antigen-presenting cells. Now, the APCs are going to take up the antigens, process them, and show them to T cells. Now, B cells do not require APCs to show the antigen, as they interact directly with that antigen, either of which both of them participate in antigen presentation. That's stage one. Stage two is the lymphocyte activation. Now remember, T cells and B cells are lymphocytes. And after antigen presentation, these lymphocytes are activated by a collection of different cytokines, and we'll get into those in a bit. T cells also will play a role in the activation of B cells. When we get to stage three, That's where lymphocyte proliferation and differentiation occur. The activated B cells and T cells will undergo many rounds of cell division to proliferate. This is what we refer to as clonal expansion. Basically, they're going to make copies of themselves. So suddenly, instead of just one particular B cell and it has an orientation to a particular antigen, you're going to have thousands of exact copies of that B cell or thousands of copies of that particular T cell. Now, some cells will be effector cells. When I use the term effector cell, it's basically those cells that engage a response to the antigen. But there's also going to be another class that's going to be made. These cells become what we refer to as memory cells. Memory cells are going to be sort of the reserve troops. They're going to be sentinels. They're going to wait out in certain lymphatic tissues. They're going to act as sentinels for subsequent antigen exposure so that when the next round of exposure occurs, they're going to start activating and activating strongly and quickly to that reintroduction, shall we say, of the same antigen or the same pathogen. Now, finally, after we've had this, shall we say, immunological war, we've got to clean up or mop up. This is where stage 4 comes in. Antigen elimination and memory cellular and memory humoral responses are going to collaborate to eliminate the antigens. Now, what you have to keep in mind here, though, is that once the threat is passed, the effector cells are going to die off. Memory cells, these are the sentinels I was mentioning a few minutes ago, are going to endure to protect against subsequent antigen arrivals. In other words, reintroduction of the same pathogen as long as the pathogen has the same antigens. You don't need effector cells hanging around. This is what helps us to not have autoimmunity situations where it's been fighting for a while and now it starts attacking our own tissues. As we mentioned before, the 3 lines of immune defense, the first and second are the innate barrier defenses and the innate cellular molecular defenses that we dealt with in Chapter 11. Now we've got adaptive defenses. This is cellular response and humoral response. That's Chapter 12. Now, as I mentioned before, and this is just a brief coverage here, when you look at cellular responses, it's all T cells. What's the advantage of that? Well, you can throw antibodies all over the place in all sorts of different tissues, but you can't get inside the cell. Humoral responses from the B cells, they're going to start kicking out lots and lots of antibodies, and they're going to be fantastic, fighting off and attaching to antigens and thereby fighting off the pathogens. But what if the pathogen goes inside the cell? That's the key crux of it. And don't just think about viruses. There are other pathogens that will hide inside of various cells. Tuberculosis bacteria, rickettsia, and a variety of other pathogens, Rocky Mountain spotted fever and all these other ones. Q fever is another type. All these organisms seem to work inside and basically act very actively inside of the cytoplasm of the cell. The problem is antibodies can't get inside to bind to the pathogen once it's inside that cell. So what we have is we have the T cells. They're specialized. They're going to find ways to knock out those cells that are infected. So we'll continue with that. Again, we go through the stages. Stage 1, antigen presentation. Stage 2, lymphocyte activation. Stage 3, lymphocyte proliferation and differentiation. Now keep in mind, the part about the differentiation is because you're going to have some cells out of these many numbers that are going to be effector cells. They're going to go right into the battle. Then we have others that are on reserve. They're going to be the memory cells, and they're going to be kept aside for further skirmishes if the pathogen shows up again, if the antigen shows up again. Now, T cells and B cells, the main lymphocytes of adaptive immunity, can recognize practically any natural or man-made antigen. Yes, that's why today we have people having allergies or reactions to foreign substances that never were in the body beforehand. Or we have new attacks that are occurring with newly evolved viruses, bacteria, etc. As I said to you before, T cells and B cells are lymphocytes. Now, the T cells mature in the thymus gland. This is kind of like a brief refresher just to keep you remembering this. B cells mature in the bone marrow. When we define the term antigen, that's any substance, if presented in the correct context that triggers an immune response. That antigen is then said to be immunogenic. Immunogenic antigens can be complete antigens. They trigger the immune response alone. Well, that's interesting, but what about haptens? Now, if you remember either in your anatomy and physiology, or if you remember in earlier parts of medical microbiology, sometimes you get these incomplete products. Think of them as almost like half of an antigen, not necessarily half, but I just want to give you it as an example. The hapten on its own is unable to trigger an immune response. So what happens is that it may require linking to a much more complex protein or polysaccharide. And in some ways, when you design vaccines, it's the same way. We sometimes attach a smaller antigenic agent, incomplete antigen, to a much larger molecule to get and stimulate an actual immune response to the particular hapten that we're looking at. Now, the term epitopes, which are otherwise referred to as antigenic determinants, that's another name for them, are really part of the antigen that the B cells and T cells recognize and mount an immune response. So think about it if you were going to say, well, if it's a triangle, it's only 1 or 2 points on that triangle for an epitope. Now, T cell receptors, TCRs. These are the antigen recognition receptors on the T cell. B cell receptors, BCR, that is an antigen recognition receptors on the B cells. I want you to get familiarized with the terminology as we get into this. Now, for example, really small molecules, penicillin, etc., they would be considered haptens. They're incomplete. They've got to have larger molecules attached to them to really trigger an immune response. But then you get into these larger molecules, lipopolysaccharides, for example, or even larger ones, the polysaccharides. Okay, so that would be an example of that would be the capsule polysaccharides that you see in Haemophilus influenzae, or even large proteins, hemagglutinin or neuraminidase proteins that you find in the influenza virions. This is where you're going from small to large, less to greater immune responses. Now, when we talk about the antigen itself, these are specific epitopes will really be specific sites where you're going to have the binding of those receptors. So it could be here, it could be here, it could be here. So it's not doesn't have to be the entire antigen. As a matter of fact, with some antigens, you will have some T cell receptors or B cell receptors that will bind to different parts of the same antigen, and they will react to those particular epitopes. Okay? Now, what you've got to keep in mind as a rule here, one epitope type recognized per lymphocyte. What does that mean? That means that the lymphocyte can't wiggle around and have more than one epitope types recognized. Instead, you will have the lymphocyte, and if it's activated, and you make many copies of those, each one of them is going to be programmed to go after that one epitope. That's where we get into antigen recognition capacity. Because really, it's unlimited. And this is due to the diverse variety of B cells and T cells that exist. In other words, coursing through your veins right now, you have a variety of B cells. Some of them will go after the same antigen, but different sections of that antigen. In other words, different sections are really what we call the epitope. So the different sections on the same antigen. They can't change their epitopes. But that's okay, because you only will need so many B cells, and if they're activated, they will make up lots of copies of that. Same thing with the T cells. Now, activated B cells and T cells will differentiate upon antigen binding to the BCR or the TCR, which I've explained. And as I said, and you see it right there, the T cells will undergo proliferation. This is referred to as clonal expansion. This is where you make lots of exact copies. Now, some will be effector cells, others will be memory cells. B cells will undergo differentiation as well, and they will become specialized. So they'll proliferate lots of copies, and then they will specialize, differentiate. Now, the two real, if you want to say, fates is plasma cells, which are these humongous cells with lots and lots of rough endoplasmic reticulum, etc., because what they're going to do produces gobs and gobs of antibodies. Now, the memory cells, those are different. The memory cells are going to be the sentinels that I mentioned before. They're going to sit on the side. They're going to probably hang out in certain lymphatic tissues, and they will be on alert for the subsequent infections that occur with pathogens that have that same antigen, that same epitope to the antigen, as a matter of fact. And see, here we have the epitope binding on the antigen binding site on your T cell. In the B cell, what they have for their antigen binding sites, interestingly enough, is basically antibody structures. Now, the antibody structures can be attached to this plasma membrane, and then when the B cells get revved up, they're going to actually freely secrete these antibodies. Okay, the antibodies where they bind, where the epitope binds to, is called the antigen binding site. So let's talk a little bit more about T cells. They fall into two main classes: helper cells or cytotoxic cells. Now, T cells are identified by specialized glycoproteins on the cell surface called cluster of differentiation proteins. You may have heard them as, CD8, CD4, CD this, CD that. And it really gives a nice fingerprint to that particular type of either T cytotoxic cell or T helper cell or something like that. We talked about two major classes. We're going to go a little bit more into T cytotoxic in a few minutes, but right off the bat, T cytotoxics directly destroy infected cells or cancer cells, in other words, tumor cells. T helper cells will coordinate an adaptive immune response by stimulating other white blood cells. T helpers really get things organized in the battle. T helpers are most abundant when it comes to the T cells. They coordinate immune responses by release of cytokines. Okay, the cytokines will boost the activity of macrophages, B cells, and T cytotoxic cells. The main organizer cells for cellular and immune and humoral branches of the adaptive immunity, well, that's T helper cells. We're going to get into the subtypes of T helper also. Now think about this for a second. When we started to have the onset of HIV that became very prominent in the 80s and 90s, what did you see? Everybody talked about immunocompromised, immunosuppressed, and things like that. The big issue was that the HIV virus would attack the T helper, which seems to be the main link to all of these other immune responses. So you break the link, you have a synchronized or deorganized or completely disorganized series of immune responses that can't get themselves together. And so now the immune system can't protect the individual against some opportunistic infections. And sadly, that's how some individuals died. They died of Kaposi sarcoma and pneumocystis carinii and a variety of other disorders. So the T helper cells are a target of the HIV virus. That's the main point I want you to walk away from. Here you see the cellular branches of the adaptive immunity here, T cytotoxic cells. They're CD8s, their CD proteins are classified as CD8. T helper is known for having CD4. And T helper is going to basically activate both T cytotoxic cells as well as the B cells, get them all revved up. But as I mentioned earlier, there are several different types of T helper cells. Now, you want to keep in mind that any population of helper cell types below will include effector and memory cells. So in each one of these classifications, there's both an effector class and a memory cell class. So the memory ones will also be acting as sentinels and will be in the lymphatic tissues on standby. The first group we have is T helper 1, TH1. They activate T cytotoxic cells, macrophages, and natural killer cells. Remember those? Those are the NK cells. So T helpers will destroy pathogens inside of host cells. The problem is that if you have excessive TH1s for a prolonged time, this is linked to autoimmunity and inflammation. T helper 2s. These primarily stimulate B cells to make antibodies. And it's been known that abnormal activity of T helper 2s is linked to allergies and select autoimmune disorders. Finally, T regulatory. Now, the T regulatory cells control the function of other white cells, including dendritic cells, mast cells, B cells, and some T cells. This is to help ensure that the immune response tapers off. In other words, after the threat subsides, the battlefield gets cleared. And so what happens is the T regulatory play a critical role in damping down all of these responses of the immune system so that it quiets down and it's all over with. No chance for autoimmune disorders, allergies, or any other issues pop up. Here you have the CD4, so this is activated T helpers. And you see the classifications, T helper 1, T helper 2, T regulatory cells. T regulatory will decrease immune responses. T helper 1s stimulate T cytotoxic cells. So you're going to have cellular response promoted. Remember, cellular response, go right after infected cells, cancer cells, tumor cells, things like that. T helper 2, you notice that they will stimulate B cells. In B cells, we have the humoral response promoted. When I talk humoral response, I'm talking about proliferation in the production of specific antibodies. Now, as you go through all of this, I know you're probably sitting there going, I got to memorize all of this. Calm down. If you see the CD markers, CD4 helps identify T helper cells. And the subclasses are T regulatory, T helper 1, T helper 2. You can see the functions of each one, which to a point I've already spoken about. Then you have CD8s. The CD8 is the T cytotoxic cells. There are subsets, but the features and roles are poorly characterized. They're still working on research into that. The interesting thing is that T cytotoxic will destroy cancer cells, cells infected with intracellular pathogens. This includes, of course, viral infections too. And the T cytotoxic populations include memory and effector cells, as all the other ones do. So, here's a question. How come my T cells and B cells don't start destroying all of my tissue randomly? Well, in the development of T cells and B cells, they are screened at the primary tissue sites that they undergo this maturation. T cells would be in the thymus, B cells would be in the bone marrow. And what are they going through? Detection for self-tolerance. Now, self-tolerance is like basically saying, okay, I see you as cardiac tissue, I see you as neurons, I see you as epithelial tissue, and I see all of your proteins. That's fine. That's not my targets. But you have to keep in mind this concept of gene shuffling. I can't remember the scientist, but he had received the Nobel Prize for finding all the different genes that basically play these roles in switching and arranging so that they will make new combinations of receptors for potential antigens. Well, is it possible that you have mathematically a chance to make potential antigens, or excuse me, receptors for potential antigens that would be for yourself, for proteins on the surface of, let's say, neurons or heart tissue or muscle tissue or anything? Yeah, you do. and if they went haywire, you'd have an autoimmune disorder. So what you have to do is you have to avoid cells making receptors targeted for self-antigens. And this is how you instill, through this filtering process, self-tolerance. Okay? What happens is, and you'll see the process, it's sort of like a yes-no dichotomous key. And what basically happens is that the screening mechanisms for T cells is in the thymus, for B cells is the bone marrow. And there are several levels of testing that occur. And if any of them have the hinting of binding to self-antigens, they're destroyed. They're just programmed to self-destruct. That's called apoptosis. Apoptosis is the term for programmed cell death. Okay? All cells are in essence being filtered so that they have tolerance for the self-antigens. Okay? Now, we're going to come upon this concept that almost gets to be a tongue twister: major histocompatibility complex proteins, MHCs. They're also been called the human leukocyte antigens, HLAs. What they are cell proteins associated with self. And what I mean by that is basically you have MHCs on cells throughout your body, two different classes of them, but most of them in one way or another have one or both. Here's the big point. They help in identification of the cell type as self. And by the way, you may have heard when people go for tissue transplants, organ transplants, things like that, they're actually looking for several MHC genes or antigens, and they want to make sure that these proteins are compatible, because if they are not, they're not a match, odds are whatever you transplant into that donor, into that recipient, is going to be rejected by the immune system. It's going to be detected and considered foreign. Okay? So here you have the T cells screened by the thymus. Do they recognize the MHCs? Yes, fine. Are they potentially self-reactive? Yes, they're destroyed. If any of this is no, well, recognize the MHCs? No, that's apoptosis, programmed cell death. If it's potentially self-reactive, no, then it passes for self-tolerance, migrates to lymphoid tissues. Same thing for the B cells. Could they make antibodies to self-antigens? Yes, it's targeted and destroyed. No, it's then passed on because it passes the self-tolerance screening, migrates to the lymphoid tissues. Now, this table is a good kind of overview. Excuse me. What you have to understand is this. You don't have to memorize all of it. Use it as a review, okay? Where you're going to find the T cells and the B cells, basically it's comparison and contrast. What do they do? What antigen recognition receptors they have? And are they considered antigen-presenting cells? And what MHC proteins are present on their surface, etc. Let's move to the cellular branch of adaptive immunity. Okay, this is where we're going to focus in on T cells. Then later on, we're going to shift over to the humoral branch of adaptive immunity. And when we talk about cellular responses, T cells mobilize against a diverse set of antigens. The cellular response is mainly organized by the T helper cells, but carried out by the T cytotoxic cells. Again, you go through the 4 stages of cellular response. Stage 1, antigen presentation. Stage 2, lymphocyte activation. Stage 3, lymphocyte proliferation and differentiation. Then stage 4, antigen elimination and memory. Excuse me. Let's start off with stage 1. Antigen-presenting cells use MH1 or MH2 to present antigens to the T cells. Remember, MHC presents antigens to the T cells. So MHC1 is found on the surface of all body cells except the red blood cells. And the covering of them provides a self-recognition. So when we talk about the MHC1, they present intercellular antigens to the T cytotoxic cells. How does this occur? Well, what happens is the cells, a lot of times intracellular antigens are chopped up by the proteasome. The proteasome is this intracellular organelle that's involved in the breakdown of cellular proteins tagged for breakdown. So if you have something that's already infected inside the cell, it'll start getting broken up by the proteasome. And then these fragments, these protein fragments of the antigen are combined with MHC. MHC will sort of form this complex, MHC1 antigen complex as it's called. And then it's going to basically come to the surface of the cell and present it to the T cytotoxic cell. Just saying, see what I found here? If you see this, this is a bad thing. And you can see this pathway here. The intracellular antigens could be pathogens, they could be toxic proteins, they could be anything. But when you have the antigen fragments, in the endoplasmic reticulum, that's where you're forming the MHC1 antigen complex. And it transports this to the surface. Okay? And that is where it will be expressed or exposed. And then you have the T cytotoxic cell that will bind to it. We'll get into that more in a minute. But what about MHC2? This is only present in antigen-presenting cells. What kind of antigen-presenting cells are we talking about? B cells, white blood cells that phagocytize, then process and present the antigens to T cells. So that will include, obviously, macrophages, dendritic cells. Actually, the most active APC in adaptive immunity is dendritic cells. And those will move throughout the lymphatic system, come into contact with something, and then begin the process. The process is that the MHC2 presents extracellular antigens to T helper cells. Now, these antigens can come from fungi, bacteria, parasitic worms. Once the APC phagocytizes the pathogen, you have then that basically a phagosome. The lysosome with digestive enzymes comes together and you form the phagolysosome, and basically the digestive enzymes break down some of the antigens. Now, you have vesicles with MHC2 that will fuse with this phagolysosome. And what the MHC will do, MHC2, is they're going to grab onto some of the antigens and they're going to form complexes. And then the MHC2 antigen complexes are displayed on the cell surface to interact with the T helper cells. That seems a lot of complexity. Just think about it this way. You're a macrophage, or you're even better, a dendritic cell. You come upon a fungal spore. Glump, you pull it inside by endocytosis. And then you start breaking it down with your digestive enzymes from the lysosome. Now, this is all basically while you're enclosed that spore, that fungal spore, inside of a vesicle. You start breaking that down, and then you have this other little vesicle containing MHC2 proteins fuse, grab onto some of the fragments of that spore that will in essence be antigens. Then the MHC2 structure with its complex will attach the cell surface like a flag and saying, hey guys, this is an invader. This is a bad thing. And it will basically teach the T helper cell, when you see this, you've got to destroy it. And you see this right here. Antigen presenting cell, dendritic cell shown here, takes up the extracellular antigens by phagocytosis. Then you have the lysosome, you have them fused together. You have other vesicles with MHC2. They will fuse, they will grab onto some of the fragments here that are the antigens, and then they will present these antigens to the exterior. They will teach the T helper cell, when you see that particular antigen, you've got to go after it. Now, table 12.3 gives you basically a nice, simple comparison. MHC1 and MHC2, okay? Of all body cells, MHC1 is going to show up except for red blood cells. The book calls it the body's uniform, sort of like you all know that they're all part of the body. Okay, fine. MHC2 is only present in antigen-presenting cells. So you're going to see these, and they're going to interact with T helper cells. That's MHC2. MHC1, though, will interact with T cytotoxic cells and for those particular CD proteins. MHC1 is basically involved with the presentation of antigens from intercellular invaders. MHC2 is extracellular invaders. Okay, we've gone through all of that. Now let's go to stage 2. T cells are activated by the antigen-presenting cells in lymphatic tissues. Okay, remember, T cell activation requires two signals. You're going to have a primary and a secondary activation signal. The primary activation signal involves T cell, TCR, in other words, that's the receptor, interacting with the MHC antigen complex. All right, so they shake hands, they start to exchange information, but there's got to be a second activation signal. We'll get to that in a second. The T cytotoxic cells only will interact with MHC1 antigen complexes. They're assisted by CD8, that T cytotoxic cell surface protein. With T helper cells, though, they only interact with MHC2 antigen complexes. They are assisted by the CD4 T helper cell surface protein. Now that we've got this sort of understood between T cytotoxic, T helper cells, and the MHC1, MHC2, that sounds nice, but we want to keep the system from misfiring, so we have a secondary backup. That backup is really the secondary activation signal. This involves co-stimulatory proteins on the APC cell surface binding. APC is going to basically bind to the T cell surface and say, this is it, this is for real. And you can see this. If this is your APC, and in this case they were using the example here of a dendritic cell. MHC1 antigen complex, they are going to interact with T cytotoxic, and you can see that link up right here. MHC2 antigen complexes interact with T helper cells, and you see this complex here. Now, keep in mind, antigen presenting cells, such as dendritic cells, can have both MHC1 and MHC2. And here is the big handshake here. But what happens is this is the primary activation signal, as you can see here, which does the first level of activation of the T helper cell. The second activation signal is a co-stimulatory proteins. One that comes from the APC, the other one comes from the T helper cell. Okay? And that is what gets the show going. Now, you got to be aware of a couple of other little side agreements or side things. One of them is superantigens. These are antigens that act as very potent T cell activators. These are a variety of bacterial toxins. They include that. And if you listen to the names, the Staphylococcus enterotoxin, the staphylococcal toxic shock toxin, and the streptococcal exotoxins. These are the ones that tend to start heading down that terrible road of sepsis, toxic shock syndrome, and other types of over revved up immune systems responding to the initial invader, but not necessarily slowing down. What happens with superantigens is that instead of MHC presentation, these antigens directly will cross-link the MHC2 and the T helper cell, and they're going to trigger a massive but non-specific activation. It is just basically as if you fired up all of your armies and they're going to just basically go throughout the land destroying everything. Non-specific activation is the key. T helper cell activation. This is where you get the result. You have the activation from the superantigens. It leads to large-scale activation of T helper cells, massive release of cytokines. Now, remember how I said this. The armies have now been activated, but they have no clear enemy. They start destroying everything around them. you'll get this massive release of cytokines, interleukin-2, interferon gamma, tumor necrosis factor alpha. What you end up with is shock, and in some cases, you can even have death as a result. Okay? Here is the adaptive immunity T helper cell, CD4. This is the MH2. But look what happens here. Bridging the gap, ignoring the specific binding site here of the T cell receptor. The superantigen binds both of them together, activates them. Okay. Now, stage 3. This is where the activated T cells undergo proliferation and differentiation. Keep in mind that those two words mean something. Proliferation, suddenly, instead of one cell, I've got 100, 1,000, 1,000,000 cells, all identical, all aimed out at one particular epitope, one particular antigen. Then they start differentiating. Differentiating is where you start going into, let's say, T cytotoxic, T helper cells, the subclasses of T helper cells, etc. Okay? In secondary lymphoid tissue, such as a lymph node, T cytotoxic and T helper cells bind to an epitope presented by the APCs. This will trigger the clonal expansion. This is the proliferation I was talking about. But then you're going to have different cytokines that are going to play a role in getting some of the troops specialized, if we'll say. You're going to have the T cells starting from that proliferation. They're going to start differentiating into T helper subclasses, as well as influencing subsequent immune responses and memory cell development. Remember, T helper 1 stimulates cellular responses. T helper 2 stimulates humoral responses. So here you have the T cell activation. Here you have the proliferation. Then what happens? They all start going to differentiate. So you're going to have T helpers, you're going to have memory T helpers, you're going to have various classes of T cytotoxic that are going to be effector. They're going to go out and destroy cells, but you're also going to have your own little batch of memory T cytotoxic cells too. Okay? And you see how these cytokines play. Don't sweat the little details here. I'm not going to demand that you know all the little interferons, interleukins, and everything else right now. But I want to encourage you that if you are very fascinated with this, there are wonderful courses in immunology that you can subsequently take. And don't be surprised that some of these become key players in defenses to either modulate the immune system up or down, depending on the pathology of the patient. Okay? Just to say that interleukin-12, interferon gamma, they will stimulate cellular responses. That allows you to get revved up, not just merely macrophages and T cytotoxic, but also NK cells. Interleukin-2 and interleukin-4 play a role in stimulating the humoral responses because you're going to act on T helper cell type 2, and that's going to rev up antibody production by the plasma cells. That is a group of B cells. Now, last section, effector T cells eliminate antigens, and memory T cells remain in the lymphatic tissues. This is where the battlefield is going to be cleared up, and you're going to have the remains left, and you're going to shut down the warriors. T cytotoxic cells act in antigen elimination. Yeah, we know that. But you got to keep in mind that you're going to have memory cells of various subclasses remain in the lymphatic tissues on guard. That's the entire key about memory cells. They are there to remain in case a subsequent attack occurs of the same pathogen, the same antigen down the road. Now, T cytotoxic cells, what are they going to do? They're going to release perforins. Perforins are proteins that form pores in the cell membrane. Basically, these proteins will fit into the membrane, create these nice pores, and you're going to have apoptosis triggered. You're also going to have granulozymes. Granzymes enter through the cell pores, and they begin to break down the cell proteins and further trigger apoptosis. Okay? That's the T cytotoxic cells. What are the T helper cells going to do? They don't directly attack the invaders. Basically, they don't directly act on the invaders, the cancer cells, or infected host cells. Instead, what these do is they will support the actions of cells that perform the immune response. So they're going to help rev up B cells, T cytotoxic cells, macrophages, and NK cells, natural killer cells. And you can see all of this going on here, okay? As you have this all revved up and the T cytotoxic cells begin punching holes in, sending in enzymes to break down, you're also going to get some of the NK and macrophages to start breaking down some of the cells as well. And, oops, that is what you have. Keep in mind that you do, after a short period of time, have to inactivate the remaining effector cells in that T cell classification. They don't hang around too long. As long as the antigen is no longer present, and they will eventually die off. The memory cells will remain, though. Now, we've talked about cellular immunity. Let's talk about the humoral response of adaptive immunity. So we've talked about cellular responses. Now we're going to talk about humoral. Humoral, we're talking almost like humor liquid. Basically, this is the term they use. When you come down to it, you just have to think about antibodies. Antibodies. Because B cells are antigen-presenting cells, but they also take a key role in sending out antibodies everywhere. And as you remember from your last subsequent chapters, as well as ANP2 in the immune system, antibodies, once they attach to an invader, that's the flag for basically all sorts of non-specific and specific attacks on that invader. There are two major types of antigens that play a role in B cell activation. There are two routes. First one is T-independent antigens. These bind to the B cells. They trigger direct activation cascade for the B cells. Usually when you see this is with repetitive antigens, in other words, polysaccharides. Now, why do we call it repetitive? Basically, it's, if you look at it almost like a sheet of paper, you'd see them all over the place on that sheet of paper because you have a bacterial capsule. It's going to have lots and lots of polysaccharides studded along on that capsule. Now, T-dependent antigens, they're a little different. They require T helper cells, especially TH2 cells, for full activation. And that's stage one. Stage two, B cells are activated by the T-dependent and T-independent antigens. The activation pathway is also two-step. First step, you have the activation signal, the binding of the antigen to the BCR. Remember, that's the B cell receptor. You want to keep in mind that B cells can act like antigen-presenting cells because B cells possess MHC-2 proteins. So they will engulf something, they will break it down, and they will present it as a complex of the MH2 and the antigen on the exterior of the cell. Okay? I should say the exterior of the cell membrane. Enough said there. The second step is the activation step. And in this second step activation, the T helper cells interact with the MHC2 antigen complex on the B cell surface. So in the second situation here, T helpers will come in, read the information, and then begin a further interaction with that MHC2 complex. The T helper cell releases cytokines that provides the secondary required activation signal. Here we have the primary activation signal here, binding of an extracellular antigen. We pull that in, we break it down into certain antigens from this larger pathogen, and we have an MHC2 antigen complex. It stays out there. Then what happens is we have the MHC2 antigen complex. It will now bind to the T cell receptor for a T helper cell. Now, what you have is the secondary activation. After you've had this nice interaction here, you end up with the T cell passing on cytokines that rev up this B cell. Now, one thing. Remember I talked about repetitive antigens? That's part of the T-independent antigens. They achieve B cell activation due to multiple binding events of the BCR with the epitope. What do I mean by that? Here, let's go over this. This is the polysaccharide. These are the epitopes of the polysaccharides studded all along this, membrane, if you want to say, or this could be for the capsule of the bacteria. But here's the deal. Because B cell receptors are going to be all around this B cell, if you have multiple contacts, multiple binding, here, 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 what's going to happen? That is going to rev up and activate the B cell. Now that we've gone through stage 2, let's go to stage 3, which is activated B cells. They're going to proliferate, differentiate into plasma cells and memory cells. Okay, proliferate again, clonal expansion. This is where you're going to have, instead of one B cell, you're going to have identical copies of lots more of these same B cells. Then some of them are going to differentiate. Some of them are going to get massive expansion in their cytoplasm, particularly for the rough endoplasmic reticulum, because that's going to be making a lot of the antibodies. And some of them are not going to take that route. They're going to actually just become memory cells. They're going to be vigilant. They're going to go off on reserve. They're going to be the sentinels hanging out in the lymphatic tissues just in case this pathogen comes back again. Now, once activated, the B cells undergo proliferation differentiation into, as I said, the effector cells. These are referred to as plasma cells or memory cells. Now, the memory cells, along with memory T helper and memory T cytotoxic cells, will reside in lymphatic tissues. I've already said that basically. You're always going to have in your lymphatic tissues some memory cells, some from B cells, some from T helper cells, and some from T cytotoxic cells. And you can see how this is. Antigen binds, B cells proliferate, lots of copies. Then they start taking routes of either developing into plasma cells, where they're going to kick out lots and lots and lots and lots of antibodies. Or they're going to take another route, and they're going to be the sentinels waiting for later on, in case there is a re-exposure. And when that happens, you're going to mount a much more quicker immune response, much faster response. Okay, finally we have stage 4. Antibodies help eliminate the antigens. No more antigens, no more need for antibodies, no more need for B cells, no more need for T cells. But until then, antibodies will bind to the antigens. Antibodies, and the name for them is immunoglobulins. They are proteins. They are secreted by the plasma cells. Antibodies have binding sites for the antigen, and once bound, they will trigger B cell activation. Now, what are the antibody functions? To neutralize antigens, to prevent them from interacting with host cells, to activate the complement proteins and complement cascades. Now, you have to keep in mind that complement cascades will lead to cytolysis, the actual destruction of the cell, opsonization, inflammation. Also, antibody functions will increase phagocytosis. How? Because what happens is that when the antibody binds, you're going to have this precipitation of small soluble antigens. Think of it this way. If your antigen is like a basketball, and that looks kind of significant, but if you're looking at antibodies as being very, very large, let's say like a ladder or a large or a moderate-sized room, you're going to have a bigger structure to go after if you have a complex that's both the antibody and the antigen together. You see, what happens is that the antibodies will go after even smaller antigens. That makes them much more detectable by phagocytes. Phagocytes see antibodies with bound structures. Doesn't matter how big or small they are, the phagocytes will attempt to grab onto them. Also, antibodies will trigger agglutination of large antigens, for example, bacteria cells. Keep in mind something. With even just these simple IgG antibodies, you've got two binding sites. That means literally that Y-shaped structure, you could have an attachment to one bacteria and then to a separate nearby bacteria. Two binding sites, each site bound to a different bacteria cell or virus or anything else. They will also serve as opsonins. These are factors that bind a target antigen to tag it for later phagocytosis. Okay, and you can see all of this here. They neutralize the antigens, so the antigens can't get in and affect the cell. And by the way, antibodies binding to antigens, the antigens don't have to be necessarily, let's say, the cell wall of a bacteria. They could be a toxin produced by that pathogenic microorganism, whether it's bacteria or fungal, etc. They will activate complement, and we've talked about that, cytolysis, opsonization, inflammation, as well as increased phagocytosis. Okay? Now, let's talk about antibody structure and isotypes for a minute. Now, there's a typo in the book. It says actually isotopes, and that's a typo. It is isotypes. A given plasma cell makes antibodies that all recognize the same epitope, okay? So what am I saying here? The plasma cell will make only one type of antibody. It will go after one particular subsection of that antigen. That subsection we refer to as an epitope. But don't fear, because lots of different plasma cells are made, and they all may also zero in on different epitopes of the same antigen. Okay, that's fine. If you take a look on figure 1221, you can take a look at the antibody structure, and it consists very simply of two heavy chains. When we talk about heavy chains, light chains, they're all protein chains. They're all protein strands. The chains are held together by covalent bonds. This is basically disulfide bonds. So you're going to see them between the two heavy chains. And then you're going to see a couple of disulfide bonds that are going to hold together the light chain to a heavy chain. All right? And then at the end of these short parts of the Y, if you'll say, just as an antibody example, you're going to have these tips, and on the tips there are going to be basically an antigen binding site, which consists of ends of both the light and the heavy chain. Okay, so you've got to have both the chains to really do the effective job of binding. Now, what I do encourage you to do is review table 12.4 for comparison and contrasting of antibody isotypes. Here's what I'm talking about here, the antigen binding site here, which would bind to a certain section of this bigger antigen, which that section we call the epitope. Notice that you have the heavy chain, 1, 2, the lighter chain, 1, 2, a disulfide bridge, covalent bond that holds the light chain to the heavy, and then several that hold the two heavies together. Okay? Now, one other thing to help you to keep in mind about isotype switching, that's a fancy term. What does it mean? B cells alter the class, the subtype of antibodies made. In other words, they can't make switch everything and just say, well, instead I'm going to go after an epitope of a virus as opposed to an epitope of a bacteria. No, you're stuck. You are committed once you've gone through this process, and you can only go after specific epitopes. But here's the good part. You can change the type of antibody made. The type of antibody will have the same binding site for those epitopes, but you can shift from isotypes from IgG to IgM, et cetera, et cetera. So what does that mean? Well, immunoglobulin G, when you compare it to immunoglobulin M, take a look at them. IgG has only 2 binding sites. IgM has 10 binding sites. Why? Because it's basically is almost if you've taken 5 antibodies, the Y-shaped little structures, fuse them together. There are some advantages to that, and we'll mention those in a couple minutes here. I want you to just bring up some basic pointers about the different isotypes. First off, most common, most abundant in human blood is IgG. It's found in almost all body fluids as well. This is the only antibody that will cross the placenta to protect the developing fetus. So in other words, mom's antibodies can cross to protect the developing fetus in utero. IgA is mostly prevalent in mucous membranes. IgM is found mainly in the blood, but it's not a strong opsonin. IgE is a monomer. It's central to fight parasites and mediates all allergic responses. So if you're doing a blood test and you look at the Ig, the antibody isotypes, and you see this massive increase in IgE, it's another type of clinical indicator for the possibility of parasitic infection or an allergic response. IgD, it's the sparsest in its availability, and no one is really clear of the precise function of it. You can see here's the IgD. Looks a little bit like a Y. IgE, the same thing. IgM, count the binding sites. 1, 2. Okay, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Usually when you have the initial infection, you're going to start producing more IgM and then shift over to lots and lots of IgEs. IgAs, you'll find them in certain secretions. Here is a comparison on table 12.4. I don't expect you to memorize all of this, so don't lose a lot of sleep. Here are a couple of important points. What I've mentioned before in the notes, what I've mentioned before in this lecture. IgG, most abundant, crosses placenta, produces later in infection. IgM comes out early in the infection. It's a large structure, but it has limits because of its size. So it can't always get into some tighter places where migration would be allowed. IgA, main antibody in milk. When you have breastfeeding of children, mom's producing a lot of IgAs, which are going to help protect the newborn. Notice that IgA has 1, 2, but in this case, it's like you've got 2 of these fused together. So you have 1, 2, 3, 4 binding sites. Okay? And I want you to get familiarized with just some of these basic points. Now, in the fog of war, the author decided to give you a little bit of notes or ways to think about things. Another way to get you clear on the differences between antigen-presenting cells, T helper cells, T cytotoxic cells, and B cells. They look kind of cute. I encourage you to review them. Keep in mind that the interaction and the proper successful function of both B cells and T cells, T cytotoxic, is in part dependent on the function of T helper cells, okay? Now, this is where we come down to this deeper exploration, and this is where we're going to get into the different aspects of adaptive immunity. First off, when we talk about memory cells, they allow for fast, amplified response upon re-exposure to an antigen. This is where we're dealing with immunological memory. Now look, this is very important because this is going to also go hand in hand with understanding and comprehension for why in some cases vaccines work, why in some cases you have to have multiple shots or a booster after X amount of period of time to maintain the immunological status. Immunological memory is basically the function of the memory cells to rapidly reactivate cellular and humoral adaptive responses if the same antigen is encountered again. See, here's the point. After the first exposure, effector cells are going to die out. You're going to have memory cells that are going to reside in the lymphatic tissues, which I've been saying many times before, but basically those are now an immune system that's been taught that this particular pathogen, this particular antigen, has a threat to the survival of the organism, and therefore it should be eliminated if it's seen again. When we talk about a secondary immune response, this will require coordinated activity of both memory T cells and memory B cells. When you have the primary exposure to the antigen, as I said to you earlier, IgM antibodies are generated first, then which follows up is IgG. Now you'll see this on a particular figure 1223. It's interesting because the secondary exposure to the antigen, instead of the surge of IgM, you're going to get a massive surge of IgG. You get lesser amount of IgMs. Now they measure the amount of antibodies present in the blood, and they have a term for it. It's called the antibody titer. For example, if you have a hepatitis B vaccination, after about 5 years, for a while, they used to say, well, what you have to do is get a booster. And I used to do that. I got a booster every 5 years. And then they started saying, well, what we need to do is do a blood test, because if you have an antibody titer over a certain amount, you don't need the booster. And then after so many years of seeing this, and what we're talking about is over a span of about 20 years, okay? When you first got the hepatitis B vaccine, it was 3 shots. First shot, then later on a second shot, 30 days, and a later shot, 90 days. Usually about 96% of the people seroconvert, meaning that they have an active immune response by the second shot. 100% seroconvert by the time they have the third shot. But then they're told, you got to come back in 5 years to get a booster. This is to prevent you from getting hepatitis B, which at the time in the 80s was quite a threat to medical care workers. Then later on, they turned around and said, well, if the antibody titers are still high, why give them another booster? And so what happens is you see this evolution of thought in the application of certain vaccines, where it might have been need to get every 5 years, to later on, well, let's test them first, and then to the point of saying, well, you're pretty much, after a couple of exposures, meaning vaccinations, you're good for the rest of your life. And that can happen. So as we mentioned here, here's the antibody titer on the y-axis. This on the x-axis is days. First exposure, IgM takes over, and as it's starting to go down, then you get this revving up of IgG. But secondary exposure, notice the incredible skyrocketing increase in IgG, and you get the ho-hum like, yeah, right, we're going over here for IgM, and then it drops down. This is the difference. You've trained the system, and so the immunological memory is there and is demonstrated by this ramping up of the resources in producing IgG antibodies, much more quicker and much more robust in their response. Now, we're going to talk for a minute about humoral immunity, and basically humoral immunity can be in several different statuses, natural or acquired, naturally acquired, or artificially acquired, and either it's active or passive. Now, I'm going to clarify each one and try to give you some sort of an example for each. I do encourage you to review this because it's very important. Naturally active immunity involves contracting the infection that triggers the immune system to make effector and memory cells to confer long-term protection. Can occur for symptomatic or asymptomatic infections. There are some people that never really get a lot of symptoms, but they are carriers of the infection, do eventually throw it off, and they have natural active immunity. Artificial active immunity is, by using vaccines, they trigger the immune response in the patient. This trains the patient's immunity to recognize specific antigens. And so what happens is you have both the effector and memory cells made, and eventually this will confer long-term immunity. So the first case, natural active immunity, I've got the measles, I've got polio, I've got chickenpox, something like that. Subsequent attacks, I would have basically long-term protection because my immune system has fought it off and now has memory cells to provide protection subsequently. Artificial active immunity, instead I get a measles vaccination, a polio vaccination, smallpox vaccination, chickenpox vaccination. So I'm training my immune system to develop an immune response for the first time, and then subsequently I have memory cells that will confer long-term immune response to that particular pathogen. Now, natural passive immunity. This is something that you're going to see. The patient received antibodies to an antigen through non-medical means. For example, the maternal antibodies in the colostrum and breast milk. A lot of times what happens is just after the baby is born, mom will begin nursing the child, and in her colostrum and later in also some of the breast milk, you will have antibodies produced. Now, there's no long-term protection. The protection only lasts as long as the antibodies are present. Okay? So as long as you get that clear. The artificial passive immunity. The patient receives protective antibodies from a medical treatment. One of the things that you hear going around right now is with COVID-19 is taking people who survived the initial illness. They have antibodies in their blood, and some of them are actually volunteering now so that some of their plasma is extracted from blood. So they donate some blood. And then basically what happens is that plasma may have sufficient antibodies to help individuals fight off the viral infection. It's very common for receiving anti-serum injections. The serum is antibodies to the pathogen or a pathogen toxin. Again, there is no long-term immune benefits. It's only short-term protection. Case in point, some people who have been exposed to individuals who have Hep A, they will get a gamma globulin shot. And the gamma globulin is usually IgG antibodies, a collection of them. Some of them will provide a short-term defense against the hepatitis A pathogen. Also, against certain snake venoms or spider venoms, there are anti-serums. And usually what they do is they take an animal, they expose them to a small amount of the venom. This stimulates their immune system, and then they extract from the blood the plasma, and then they extract from that the antibodies and use that as a treatment. It's very common for poisonous snake venom, rattlesnakes, for example, cottonmouth, and in some cases the coral snakes. Those are common in the North American area. What they'll do is they'll just have the anti-serum for that at hospitals, so the individual will take it and they will survive as opposed to dying from the snake venom. So I would encourage you to take and review each type and consider an example for each one of these different types. So is the process natural? Yes. Naturally acquired immunity. The active acquired immunity would be from a previous infection. If it's a naturally acquired passive, it's antibodies passed across the placenta or through breast milk. They only last as long as the antibodies are there. What you really want is a complete active immunity, which means that you're going to have memory cells that are going to last for a much longer period of time. And those memory cells are not only for B cells, but obviously, of course, T helper cells and memory T cytotoxic cells. Artificially acquired, if you have artificially acquired active immunity, that's usually an example is vaccinations. Artificially acquired passive immunity, that's usually something like anti-venom. okay? Basically antiserums that neutralize toxins. Okay, we're pretty much finishing up here. I would encourage you to review the visual summary on page 373. That will include some parts of a review for the innate immune system too, so don't panic about that. Review chapter overview of page 374. Review the end of the chapter questions 376, 377. Review also homework in Mastering Microbiology, as well as the quiz in Mastering Microbiology. Peruse the videos in the Blackboard for the lecture content, and the next section we will be going into is immune disorders, which are chapter 13. Have a wonderful day.